Excellent. Welcome to lesson 13 on year 3 of science. And we have the lesson on fungi and protista. So this is a very, very interesting lesson. Okay. And the first thing that we want to do, okay, is to think about, okay, here in the attention activity, it talks about comparing different types of bread. What is the difference between bread and a tortilla, for example? What would the difference be between one and the other?
Bread has yeast, so it rises up. Then tortilla is just flat and it has zero yeast, and yeast makes it rise up. But since a tortilla doesn't have any yeast, it just remains flat. They're still really good.
Yeah.
Yeah.
That's right. Yes. So the tortilla has no yeast, okay, and the yeast is what makes the bread rise, okay. Other than that, the recipes are pretty similar, okay. So today we are going to talk about some of these organisms and microorganisms, and yeast is one of those organisms that we are going to talk about. Now, to begin with, we are going to see that there are different types of microorganisms. And here I have a link to this Microbe World website that is really cool. And it tells us that there are 6 different types of microorganisms: archaea, bacteria that are prokaryotes, fungi, protista that are eukaryotes, viruses that are neither. So viruses is interesting. We are going to have a whole lesson on viruses, but they are not classified really as either. People don't even know whether to classify them as being alive or not, okay? And then we have these microbial mergers, okay? So this is one of the few sites where I heard this classification of microbial mergers. And it's not that they are special types of microorganisms. This is basically saying that there are 2 types of organisms that join together in order to do something together. So it's more of a symbiotic relationship. Aubrey asked, are fungi or viruses the most common or neither? Okay, so this is an interesting question. Okay. I would probably say that bacteria are most common than anything else. Okay, there's bacteria literally everywhere. So in some website I was reading that if the human body has 100 million cells, okay, so if we have 100 million cells, there are 1 trillion bacteria inside of a human body. Okay, 1 trillion bacteria. So as far as numbers, they are tiny, they are very, very small, okay, but there's a lot of bacteria. As far as number goes, I think that bacteria take the first prize, okay. Connor wants to say hi to Sandy real fast. Okay. You said hi. All right. Fungi are also everywhere, and protista are also everywhere. They are larger than bacteria. Remember that archaea and bacteria are protista, and fungi and archaea and bacteria are prokaryote, okay, and fungi and protista are eukaryote. I unmuted you, Josh. And viruses are neither, and these microbial mergers are just kind of a combination of maybe a bacteria and a fungi, or a plant and a fungi, or an animal and a bacteria, and so on, okay? This website is really cool in that it has these links where you can go and study more about each one of those microbes, okay? And so we have here the different links, okay, that says how are they different, classification, and so on. So it's pretty accessible, I would say, all right? With fungi, they have also that information with different list of things and so on. Okay, so those are the different types of microbes that are around, all right? So in this lesson, we are going to talk about fungi and protista, because those are the last two of the eukaryotic groups that we are going to study. And then in next lessons, we are going to do bacteria and archaea. We have a lesson on viruses and so on, okay? One of the things that I wanted to mention was that, let me see, what was it that I wanted to say? I had it just in my mind, and it kind of went out of my mind. Oh, well, it may come back. Okay. All right. So let's go. Oh, this is what I wanted to mention. Fungi, because they are really a kingdom in itself, and protista, that is actually also you know, a whole group of different kingdoms, very diverse type of grouping. You could literally spend a whole lifetime studying each one of these, okay? So we are not giving them near enough attention as what we could. There are people that dedicate all their life studying fungi, for example, and it's fascinating, really. They are called mycologists, all right? And so, you know, here we are just going to touch a little bit on them. So let's see, Emmeline, could you unmute yourself? Let me see if I have you. Did I unmute you? No. Let me unmute you. And Emmeline, okay, would you like to take us here through academic concept 2 and tell us what you have learned about fungi, please?
Yes. Okay. So I love this. Okay. Let me just say that first of all. But the academic concept for 2 is fungi are the main decomposers of organic matter and are very useful to plants and animals. So first of all, it kind of goes through yeast, which, as we have learned, is a microbe and is a single-celled organism, and how it needs energy, produce waste, and it produces carbon dioxide. And it kind of lists several different functions, like cellular respiration that it can do, and oxygen when it has oxygen, and fermentation, and it also mentions that it's unicellular and reproduces by mitosis or a budding process. And at first I was kind of like puzzled. I was like, so what is like a budding process? And what kind of happens in the budding process is that it's a form of asexual reproduction. So technically fungi or fungus can do 2 types of reproduction, both asexual and sexual. And budding is a form of asexual reproduction, and it's when a new organism develops from an outgrowth or bud due to cell division at one particular site. So you look at the picture that's on the screen, and you have one big body and one small little body. And what's happening is that the small little body is trying to branch off, but it won't fully depart from I don't know what you want to call it, the mother cell, until it is completely developed. And then it leaves, and it kind of leaves like interestingly enough, a scar tissue, which I thought was like so awesome. So that's kind of what happens when budding comes. But it also talks about molds, and they're very common and diverse, and they're multicellular, and they grow as a network of tubular, branching, interconnecting cells called hyphae. Is that right? Hyphae?
Hyphae, yeah.
Okay. And that basically is a long branching filamentous structure, and it is the main mode of vegetative growth and are collectively called mycelium. Is that correct? So you look on the screen, and this kind of like long branching line is basically hyphae. And the next picture is basically talking about all the different types of things that you can find. So number one would be like the hyphal wall, so it's kind of, just protecting the outside of everything. And then you have the septum, which is a partition dividing filamentous hyphae. So we've talked about the hyphae into discrete cells. And then you have...
This is really interesting, Emmeline, that the cells, so this is one cell and this is another cell, but they are not completely divided.
Yes.
In the hypha. The cytosol and the nuclei the mitochondria, all the organelles can move from one cell to the other. Isn't that amazing?
Yes, it's super interesting. I was like, oh my gosh.
Yeah, So really, really cool. Keep going, sorry.
So again, just kind of, I don't want to list all of them because we probably won't have enough time to go over all of them. But you have some of the ones that we've kind of gone over, the mitochondrion. They have vacuoles and ergosterol, which is basically a sterol found in cell membranes, and it serves kind of like cholesterol in animal cells. So the purpose and function is kind of similar to that. And then you have the ribosome, the nucleus, the endoplasmic reticulum, the lipid body, plasma membrane, and correct me if this is right, is it pronounced Spitzenkorper?
Yes.
Okay. And that is basically organizing the hyphal growth and the morphogenesis, which I think we've gone over morphogenesis, but if we haven't, it's basically like how the cell forms, like gives it shape. And then you have the Golgi apparatus. But yeah, it's pretty awesome, and that pretty much sums up that. But another thing that it goes on to is how fungi decompose or break down dead plants and make medicine, like penicillin, and also they cause diseases and gives yeast as an example. Provide food. I don't know. Is there anything you want to add, Mr. Boldy? I feel like I'm going on a tangent.
Anna is asking, what do you use penicillin for? Do you know?
You use penicillin. It's it's
so penicillin is
like infections and diseases, right?
Yeah,
right?
it is bacteria. Okay. So there's a fungus called Penicillium, okay, that produces an enzyme called penicillin, okay, and penicillin actually breaks down the cell wall of bacteria, and so once the cell wall is decomposed, the bacteria just kaput, okay. And so it is actually a very, very important thing. We are going to talk we have a lesson where I'm planning on talking specifically about penicillin and how it was discovered. It is an amazing story, really, and it is one of those, frankly, a gift from God. Okay, I can tell you that probably every single person in the world that is alive today is alive because of penicillin, one way or the other. Okay. I, for example, would be dead if it weren't for penicillin, because I had a bacterial infection when I was 10 years old. And the only thing that kind of saved me, it was penicillin. So really, really interesting. Okay. Yeah. So let's switch now. Okay, pretty soon we are going to talk. Okay, about now we are going to switch to Haley. Do you want to do the? Oh, I just click off the last question. I think that Anna was asking the question. So fungi are really, really amazing on these things. Emmeline, a couple of questions. Where do the fungi grow from? All right. So, for example, we have here the hypha. All right. At which part of the hypha do they grow? Does the fungi grow? Do you know? Emmeline? Okay. So the hypha, let me answer, okay, my question. The hypha basically grows here from the tips. And so it branches out and it makes a huge network of this filament. And it is really amazing that the extent at which the mycelium, which is basically the whole collection of a hypha of an organism, can grow. There's one mycelium that is the largest organism on the earth. Okay, it's a forest. It covers a whole forest in eastern Oregon, and it's one mycelium. And the mycelium just decomposes the forest and lets new forest grow, and then it eats it back again, and so on. But it's about, I don't know how many, acres. I don't. Oh, okay. Here we have. So it's 2,400 acres, and it is estimated to be 2,200 years old. That is one mycelium. Okay. So, yeah, and fungi are really a kingdom. Okay. All right. So Connor is asking, what's wrong with the guy's hand? So there are some fungi that grow in different areas. So let me go over to that one. Okay. So let's see. We have that. Let's see if we have the picture there. Okay. So there are some fungi that grow in different areas. One of the type of fungi can grow in the skin. And so a very common place for getting a fungal infection is between the toes. That is called athlete's foot. And it is very, very hard to get rid of. It sometimes is kind of painful because the fungi attacks the nerves, okay, and also breaks down the epithelial cells, the skin cells, and makes these kind of cracks on the skin. So this is actually a fungus growing in the toes of a person, okay? So That is what is happening. So fungi are heterotrophs, meaning that they eat from other organisms or they eat other organisms. Like this one here, it's eating from the tree, or they decompose material. But it's actually a very, very interesting and important thing, because imagine what would happen, okay, what would happen without fungi? What would happen, let's say, to a tree when the tree dies, okay, and it falls to the ground? What would happen with the tree if there were no fungi? What do you think that would happen? Liberty says it will never break down. Okay. Yeah.
I have an answer.
Yes, go ahead.
Take up lots of forest space, not allowing more regrowth. So by this period in time, if there were no fungi, it would just have a ton of logs lying around, and there would be no trees left. It just
That's right.
Grow for all the space problems.
Yeah, yes, So fungi are absolutely important in decomposing organic matter. So they are actually very, very useful. If not, we would never be able to recycle organic matter. Aubrey is asking, does moss sort of represent fungi or like another name for it, or are they completely different things? So we are going to take about mosses, okay, in now the next concept. Emmeline says, furthermore, without fungi, trees wouldn't be able to get phosphorus nutrients. Could you explain that, Emmeline? You can come back on the mic. All right, what do you mean by without fungi, trees would not be able to obtain phosphorus nutrients? Go ahead. Okay, I cannot hear you. Your mic is open, but I cannot hear you, Emmeline. So check to see if your sound is up or what. Okay. So it says you are muted. I am. Let me try again. So you are unmuted. Okay. All right. So, I had you before. Okay. So let's move on. All right. So the idea is that many trees, in fact, most plants, most vascular plants, they form a sort of relationship between the plant and a fungus. Okay. So let me see where it is. So I think that it would be over, not this one. So the idea is that they collaborate. So for example, this is the root of a plant, okay? So let's see, Emmeline says, what happens is that the trees give fungus sugar, and the fungus gives trees nutrients like phosphorus that are scarce. That's absolutely right, Emmeline, excellent. Okay, then through this transportation, it goes back and forth. That's why fungi are around the roots, absolutely. So the fungi, they help the roots grow. The fungi, let me see if I can show you. In the hyphae, the fungi secrete, especially here in this area, okay? In this area, the hyphae secretes enzymes that are able to break down matter. And so that is how the hyphae makes room for it to grow, is by breaking down what is in front of it, all right? And it can break down things that the roots cannot break down. And so the roots kind of plants follow behind the hypha of fungi, and they are able to penetrate rocks and soil and obtain phosphorus and nitrogen and so on that the plants need, but they cannot break down themselves. At the same time, the plant, the roots of the plant, give to the fungus, okay, carbohydrates. So the plant produces carbohydrates up in the leaves by photosynthesis, and they can send those carbohydrates down to the roots, and they feed both the roots and the fungi too. All right, so it is really, they work together. It's really an amazing thing. Okay, so let's go now to academic concept 3 on Protista. So thank you, and let's see, Haley, would you like to tell us what you know about Protista? Do you have a mic, Haley? Do I see Haley here, or did I scare Haley away? I hope not. I'm in trouble now. Uh-oh. Uh-oh. Okay. Oh, I did that. Okay. Oh. Okay. Emily's mic is not working. We lost people. Okay, so anyone that would like to no, Connor says, anyone that would like to talk about Protista really fast, 5 minutes I have. Connor, would you like to, please? Griffin, would you like to talk about Protista? Okay, no problem. Okay, so I hear a mic. Yeah, no problem, Connor. Okay. All right. So I'm going to talk about Protista because I think that they are really cool. All right. So I get the 5 extra points. None of you do. Ha! There. All right. So Protista is the other group of eukaryotic organisms, but they are not really closely related. Okay, they are basically, I like how Mr. Anderson in his video here, he compares them, you know, to basically being, you know, let's see, how does he put it? A whole, you know, drawer full of stuff, okay? So it's all the eukaryotic cells, all the eukaryotic organisms that are out there, that are not plants, they are not fungi, you know, and so on. Anna is asking, protists and Protista are the same thing, right? Yes, they are, okay? In fact, protist is the more common term now. Protista is kind of an older classification. Okay, people keep changing the names of these things when they come up with different ideas, but that is basically how it goes. All right, the main characteristics of protists is that they are not of one descendancy. They are all eukaryotic. Some are multicellular or unicellular, and there's no tissue differentiation. Okay, so there are some that are very small, that are microbial, and some that are very large, that are also protist. So, for example, kelp is protist, all right? So have you ever seen a forest of kelp? In the ocean, there are some parts where is a bunch of kelp, okay? Mosses are also protist, all right? So that is, Anna, I think, that was asking about that. Chas says that he eats kelp, okay? Where do you eat kelp, Chas? see the kelp, how it grows, and the interesting thing, go Patrick. Patrick is going to check his results one last time, okay? Not kale, but kelp, okay? It's an algae, and it grows in the water, and the interesting thing is that kelp, even though it's really large, it doesn't have differentiated tissue. It's all of one same type of tissue. Okay, so that is why it's classified as a protist. Does that make sense? And so these are here the forest of kelp. This is a different organism. All right. Yes. So they are very, very large. What is that white thing? Okay, so let's go back to the previous picture. This white thing is an anemone, all right, and that is a different organism. It's an eukaryotic organism, but it's not a protist. Okay, all right. Yes, Nathan, you are going to report on your experiment pretty soon. Okay, all right. So sometimes if people are swimming in this kelp forest, they can get trapped, so you need to be careful, all right? Nemo lived here in an anemone, all right? Okay, yes. The anemone don't sting, okay? They kind of do, but they actually, when you touch them, they emit a little electroshock, okay? And so they paralyze, if small plankton or a small fish, and when they touch the tips, they give a little shock that paralyzes, and then they are able to catch and feed themselves to that. Okay, all right. So excellent. So protists can be very large or very small. And like I'm saying, there are some that are microscopic, really. Okay, let's see what else can we see here. Some do photosynthesis, and others eat other organisms. And here we are going to see really fast a very cool video of an amoeba eating two paramecia. Okay, paramecia is the paramecium is also a protist, and the amoeba is a protist. So here we have one type of protist eating another type of protist. Okay, and that is not abnormal. You have lots of animals eating each other. Here we have the amoeba. All right, let's see. Let's stop here and ask a couple of questions. So let's see what we see here. Okay, so let's see. Connor says that he ate kelp chips. Okay, all right. So let's see if I can keep the question panel up and start the. Okay. The amoeba also can eat bacteria. Bacteria would be a lot smaller. So the paramecium here, the paramecia, are eukaryotic. Okay, so you see here the cell nucleus of the paramecia, and this is the amoeba here moving through. What is the amoeba doing? What is the amoeba doing here? Okay, so I'm going to have Nathan and Patrick presenting the results of their experiment pretty soon. So the amoeba here is extending, can you see how it is extending the cytoplasm around the paramecia? Okay. The paramecia at this point is not noticing anything, all right? Okay. And the interesting thing is how put together the things that we have already studied, about cell movement and all that type of thing. Okay, lysosomes and vacuoles and so on. Think about how is the amoeba moving and extending itself around the paramecia. Okay. See, now it's totally surrounded. So it's going to close here the membrane around the paramecia. So let me tell you what is going on. The amoeba is taking in water in order to grow and is moving the cytoskeleton. Remember the cytoskeleton that we talk about, that network of filaments and everything, and that is what is moving in order to enclose and extend itself. All right. So this is not in our body. This is in water. Okay. So yeah, it is like a snake going around its victim. Yeah. So it's going to get captured, the paramecia. But see what happens here. Okay. Now it is all enclosed. Okay. Now, why do the paramecia react once they are enclosed? Before, the paramecia had no idea of what was going on. Once the cell membrane here encloses itself around in the amoeba, this is now what type of organelle in the amoeba? What organelle is this now? You can type your answers or unmute yourself, okay? What type of organelle is that now?
Oh, this was my mind. Isn't it portraying the metabolism?
Patrick says a vacuole, and Anna says a lysosome. Okay, so both are correct. So a vacuole, because it's now just a large empty space, okay, and a lysosome, because as soon as it's enclosed, the vacuole is formed, the amoeba starts pumping into the vacuole hydrogen in order to increase the acidity. Okay, remember that the lysosome is where the cell digests things. Okay, and it does that by making it acidic, making the environment very acidic, and then pumping enzymes that are going to break down whatever is inside. Okay, so let's watch that again. Okay, so let's watch that part again. As the amoeba moves in and starts enclosing, see, as soon as the cell wall encloses, then it starts pumping hydrogen, increasing the acidity. At that point is when the paramecia realize, oh, I'm in trouble. This is no Kansas anymore, okay? All right? So that is when the paramecia start going nuts, okay, and jumping all around, okay? Paramecia, by the way, usually they move really slow. They move just with the cilia around them, very slow. But once they are detecting that the environment change, that is getting very acidic, they start jumping like crazy. Now, what is going on here? What is going on, guys? What are you seeing? Connor says they are combining. It's not that they are combining, they are dying. Okay? It's being digested, Patrick says. Okay? They are melting. So what happens is, remember, in the amoeba, makes the vacuole, turns the vacuole into an acidic lysosome, and then it starts pumping enzymes to break down what? What do you think that those enzymes are going to break down of the paramecium? What are they going to attack in the paramecium? That is going to attack the cell wall and the cell membrane of the paramecium, and it's going to break down everything in the paramecium, and that then is going to become part of the amoeba. Okay? Does that make sense? Okay, let's watch that again. Okay, so here we see how the amoeba is really digesting, okay, the paramecium. Now, all the things that were inside the paramecium, all the proteins, all the phospholipids of the membrane of the paramecium and everything are going to become available to the amoeba. What do you think that the amoeba is going to do next, okay, after having so much food? What do you think that the amoeba would do next?
Go work out.
Go work out? Explode? I don't think that it's going to explode, okay. Throw up? No, I don't think so. What do you think? Remember, okay, we talk about Samuel says grow. Excellent. And after grow, what happens with cells after they go through the growth period? Give to other cells? They would reproduce. Emmeline says they would reproduce. That's right. So now, after eating so much, now the amoeba will have enough material to start into the S phase. Remember, we talked last time about reproduction, about cellular reproduction. So if eating here represents growth for the cell, after that, the amoeba is going to be ready to go into the S phase to duplicate the DNA and then do cellular reproduction. Okay? Fantastic. All right, so now I would like to turn the time to Patrick and to Nathan, okay, to share the results of their experiments. Okay? So who would like to go first? Nathan, are you ready to share the results of your experiment?
Yes.
Go ahead. Okay, Patrick next. Mm-hmm.
So my hypothesis was that the bowls with more sugar would grow longer, grow taller because of the supplement in it. And then as I did it, the control one didn't grow at all during the time it did. It grew a little bit after the very end, the one with 0 sugar. So I kind of guessed that, but then the one with the one with a 1 teaspoon of sugar, there's one with a 1 teaspoon of sugar, 2 teaspoons, and 3 teaspoons. The one with the least amount of sugar, so the one with the 1 teaspoon, grew the farthest. And it first grew, well, after 10 minutes, the first one grew about 2 centimeters. And then the second one, the one with 2 teaspoons of sugar, grew about like 1.5, and then the one with the 3 teaspoons didn't grow that much. And I thought I was odd, and so did my mom when she was helping with this. She said I thought that the more sugar it would do, the more it would do, the more it would grow. But that wasn't the case. So then after 20 minutes, the first one had grew 12 millimeters, and then the second one has grown 2 centimeters, and the third one has grown 1 centimeter. So I kept on finding that odd that would do that.
Interesting. Yes, I think that Patrick has similar results. Patrick, okay. So let me tell you guys what is going on. So one of the assignments for the class is to do this experiment, okay? And Nathan and Patrick have done the experiment beforehand, before the class, and so they are sharing the results with us. So Nathan, you did just one round with 4 samples, one with no sugar, then another one with 1 teaspoon, another one with 2, and another one with 3. Is that correct?
Yeah, but I also did it with honey.
Okay, so you did it twice. You did it with regular sugar and then with honey. Okay, go ahead.
This honey was pure honey, but it was kind of really old. So I don't know if that would affect or anything, but the same thing happened that it grew more, one with a less amount of honey, than the one that had more honey. So
interesting. So that is kind of counterintuitive, because your hypothesis was that the more sugar it would grow more. Okay.
Yeah.
Yeah.
That's what I thought, and then of course, then once we found that happened, that totally changed my height. That totally went against what I was thinking and what everyone else was thinking in my family. So it was very interesting.
Yes. Let's see what are the results that Patrick got. Okay, Patrick. Thank you so much, Nathan. Very, very interesting.
I just did this actually. A huge mess in the kitchen. Don't tell my mom. So I did no sugar or sweetener, 1 teaspoon of sugar, 2 teaspoons of sugar, 3 teaspoons of sugar. Try saying that 10 times fast. And then I did 1 teaspoon of Truvia, 2 teaspoons of Truvia, and 3 teaspoons of Truvia. It's an artificial sweetener. I don't know if you guys have that, but it's pretty big here in Wisconsin. And
okay. What is it called? What is it called? Type it in the question panel. Okay. All right. Truvia. Okay. I haven't heard of that, but it's an artificial sweetener.
Yep.
Let's find out about it.
The no sugar actually got up to 5 centimeters. It grew 2 centimeters after it started rising over the course of an hour, checking it every 20 minutes. The 1 teaspoon of sugar only grew like half of a centimeter, and the 2 teaspoons of sugar grew 1 centimeter. The 3 teaspoons of sugar barely grew like 1/4 of a centimeter. It was horrible. Almost nothing grew. And then with the Truvia, it was a tie the entire time. No matter how much you put in, it didn't make a difference. It was all
the Truvia was exactly the same, so it didn't make a difference.
No, it was all 4 centimeters.
Interesting. And your results with the sugar actually then mirror the results pretty much that Nathan got.
Yeah.
Very, very interesting. So it kind of counterintuitive, because I would have also thought that the more sugar, it would grow more. So let's think about why. Why would it be okay?
I didn't know it was made by Coca-Cola.
See, interesting. So Truvia is a stevia-based sugar substitute developed jointly by the Coca-Cola Company and Cargill. Okay. So we could probably read more, but so apparently, from what you got, the results that you got, the Truvia, the yeast, okay, cannot eat Truvia.
No, I guess not.
Okay, so basically it cannot metabolize the Truvia. All right.
It says nature's bounty on the box.
Yes.
That's pretty funny.
Well, we would need to find out what the heck is made of. All right. So Cargill is a company that makes seeds, all right? And so they produce a lot of grains and everything. So it has to be organic, but it would be interesting to find out more about how is it composed, okay? Aubrey says it's funny that I didn't think what Truvia was. Well, sorry, there's a lot of things that I don't know, okay? So let's see. So it seems the interesting thing is that it seems that with the sugar, the yeast didn't eat the sugar also. What was in the control group, what was the yeast eating then without any sugar? What was the yeast eating? Because it did grow.
Probably
okay.
the water. It was probably just sucking up the water because it was gosh dang it, space vacuums. No, not that. Dehydrated. There you go.
Or it could have been
so,
sugar flour.
Yeah, the flour, you said, Nathan?
Yeah, it could have been that, but when we made bread, by the way we make it, we put the flour in and then the yeast, and then we put water in, and then that's how it gets all poofy. So it could be doing the water and the flour. And it depends on like how much you put in the yeast, whether or not it's more the flour or more the water.
That's right. Yeah. So very, very good. So the water certainly is necessary. Okay. So we have already learned, especially in a dehydrated yeast, that in order to kind of come back alive, it will need the water. Okay, to hydrate itself. But what it consumed in order to feed itself, it was the carbohydrates in the flour. All right, so remember that flour is made out of the starch of wheat, and that starch is a sugar in itself. It's not a very sweet sugar, but it is a sugar. It's part of the carbohydrate group. Okay, so the table, what am I saying? The regular sugar, okay, what we would call the table sugar, okay, is, let's see, so the crystalline type of sugar that we see commonly is actually, it says here, is composed also of carbohydrates, but it is a disaccharide, okay? So sucrose, which is table sugar, is a disaccharide. It's composed of one molecule of glucose and one of fructose, all right? So that is the crystalline table sugar, and apparently the yeast didn't go much for that, okay? So it preferred the sugar in the starch directly, okay? So if we go to starch, is a carbohydrate consisting of a large number of glucose units, all right? And as we have seen when we were talking about cellular respiration and fermentation, that is the number one, glucose of choice, sugar of choice, glucose directly. Okay. All right. So Chad is asking, you made acid. Okay. So not in this case. The yeast was doing cellular respiration and producing what makes the yeast, the bread rise in the yeast. What is actually doing that, Patrick or Nathan? What does it make it rise? What is the bubbles? Aubrey is basically thinking that maybe the water was with some impurities and so on, and very well that could be, but that is not the problem, okay? So even clean water still has some bacteria or some protists in it. Okay. So what makes the bubbles of yeast? Okay, when you mix the yeast with what was it making grow here? It makes a bunch of bubbles. What are those bubbles made of? Anyone? Patrick says carbon dioxide. Yes, that's right. So it's the result of cellular respiration. Connor is asking, says, if you take a soda and put a thumbtack on the side, why does it explode? I have no idea. We will try that in the physics class, I think. Okay. So Aubrey says it's alive and it's becoming active. So that's true. The yeast is becoming active and is actually eating the sugars in the starch and producing, as a result, carbon dioxide. And the bubbles are of carbon dioxide, okay? So when you cook the bread, okay, all of that space in the bubbles, okay, is carbon dioxide inside. Of course, we don't taste it or anything, you know, so, okay. Aubrey is asking if it makes alcohol. Do you know, Patrick or Nathan, when do yeast make alcohol? Or anybody? When do they make alcohol? Patrick says when it ferments. That's right. So if you let yeast for a long period of time, is going to use up all the oxygen and then is going to start doing fermentation instead of cellular respiration. So if you do this experiment that we have here in the class, you know, it's just a 20-minute experiment. You are mixing everything. There's still plenty of oxygen here that the yeast is doing respiration. But if you leave it, you know, maybe 12 hours or so, then it's going to start doing fermentation, and that is making alcohol as a result, carbon dioxide and alcohol, and that is what gives the sour taste of sourdough bread. Okay, so the sour part of the sourdough bread is the alcohol. Aubrey says that she's going to make wine as an experiment. Excellent, yeah. So when you leave grapes in a closed container in a hot car, it smells like alcohol. Why? Why do you think, Audrey? Okay. Yeah, why would the grapes start smelling like alcohol? And let me see, I saw a picture here that was pretty cool. Okay, so I saw something that was showing, let me see, okay, I'm clicking everywhere. Fungus. Okay, so there was a picture in one of these articles about, so the grapes, okay, I don't know, I cannot see it, but have you seen, you know, in grapes, when you buy grapes from the store, sometimes they have like a white sheen on the skin, okay, have you seen that, you know, and it's in a lot of fruits, all right? Well, that white sheen is actually fungi on top of the skin, okay? And it's fine. You can eat it. It's not that it's dirty. It's just fine. But when you leave the grape out there, especially on a hot place or something, all right, you are going to, the yeast is going to start fermenting, okay? So Connor is reminding me that we are done with time. We started late because I was asking for volunteers, okay? And I'm going to leave Anna. I owe her time for doing the gospel principle for that, but I thought that this was actually fascinating. Anna, would you like to leave us with a word of wisdom on the gospel principle related to this lesson, if you are there? Okay.
Okay.
Thank you, Connor.
So the gospel principle is about how a little leaven lifts the whole loaf, and we can be the Lord's leaven in the world. So just what we were talking about, how yeast makes bread dough get puffy and it rises the bread. Just like that, we can have a positive influence on those around us and help to lift them up. And it gives the example of King Benjamin and how he was talking to the people in Zarahemla, and his words caused the people to have a mighty change in their heart and to want to do what's right. And then it asks a couple questions in here about people throughout history that have had a tremendous positive influence on the people around them, and then people that I know. So kind of from history, I thought of a couple examples like George Washington and President Monson and Marco Polo, and I think we learned about in history a couple weeks ago about a minister who helped a village during the Black Plague to help each other. So I thought that was really cool. And then examples in my life would be, we had this missionary in my ward a couple months ago who was just really friendly and nice to everyone, and she had a lot of positive influence on me and everyone around her. So those are just some examples that I could think of.
Excellent, excellent. So I really like that example and the relation here that it makes with leaven, really. So I thought that was very, very appropriate. One of the assignments, so the assignments for this lesson, they are here, okay? One of the assignments would be to do the experiment, okay, like Nathan and Patrick did, or to do kind of the write-up of the amoeba's lunch, or to write a letter to someone that has been an influence in your life, okay, or that you think that you can be an influence to them, all right? So, excellent. Thank you very much, Anna. Andrew, would you like to dismiss the class with a word of prayer, please?
Yeah, can you hear me?
Yes.
Alright. Dear Heavenly Father, thank you for this day. Thank that we could come to class and learn something new, and please bless that we'll do all of our assignments, and please bless all the missionaries in the mission field, and say these things in the name of Jesus Christ. Amen.
Amen. Thank you guys for your participation. Okay. Emmeline, I'm so sorry that your microphone went down. I don't know what happened. So I'm going to stop the recording.